Okay, well, thank you for being here today. Like I was just told, I'm going to present to you my, fairly recent project and data. Most of it was worked before I even joined this university while I was a postdoc. Oh, I'm sorry, I'm hearing myself as an echo. I think I should be on the Zoom actually. All right, that's probably better. No, it's not. Okay. There we go. All right, perfect. Can you still hear me clearly? Yes, great. Okay, so today I'm going to talk about this project, epigenetic erosion of histone H4 lysine 20 methylation, specifically monomethylation that's induced by inflammatory molecules and inflammation that's related to aging, and how that actually leads to a very unusual form of cell death that is called ferroptosis. You can see here in the background, actually, the model that I use, which is skeletal muscle. Most people assume it's lava for some reason. It's just a cross-section of mouse skeletal muscle with laminin in red and those beautiful stem cells in green over there. So I'm going to start by introducing skeletal muscles. Of course, skeletal muscles represent 40% of your body mass. They are useful for motility, breathing, something that we don't really appreciate every day in our life, but you do, need them to actually live a good quality of life. And what's really incredible about this tissue is that it has the incredible ability to regenerate following an injury. And when I say injury, you might think about, a car crash or some acute injury that happens, but it's also on a daily basis when you hit the gym or go for a run and you have micro tears in your muscle, you need to regenerate your muscle fibers. And this process is mostly carried out by resident adult stem cells that are called satellite cells or muscle stem cells. They're called satellite cells because of their location. You can see it here on this isolated single muscle fiber that is multinucleated because it comes from the formation of multiple myoblasts. But the single cell is the supportive niche of their stem cell that are PAX7 positive. It's something that I'm going to use throughout the presentation. And you can see it here again in this cross-section of a muscle between the fibers. And so at homeostasis, those cells are called quiescent, which means they're in dormant state to preserve their stemness and their activity from external cues. In response to certain cues, such as damaged fiber, they will go through a process called activation. Will they re-enter cell cycle, self-renew, decide whether they want to go back to quiescence, commit into a myoblast, where they will proliferate and differentiate into regeneration. And this entire, cell fate decision is very well characterized and regulated through a genetic cascade of transcription factors called the myogenic determinant factors that initiate with PAX7 when the cells are actually in quiescence, all self-renewing. So those are the true stem cells. Upon activation, PAX7 will activate the transcription factor MIF5. MIF5 will then activate MyoD. At this point, the cells has a choice whether it turns off PAX7 and continue toward terminal differentiation by activating myogenin, or it can go back to being quiescent cells by shutting off MyoD and being back to only having PAX7 expressed. And so the real interest in my lab is to focus on how those processes and muscle stem cell function and regeneration is altered throughout aging. Same thing, it's very well documented during aging, whether it's in mice, which is the model I use, or in humans. As you grow older, the number and the function of muscle stem cells decline with time, which means that even though not only you have less muscle stem cells as you grow old, the one that survive and remains are actually also less functional. And that is due to a collection of extrinsic and intrinsic changes that we're going to talk about right now. I mentioned also that this is associated with sarcopenia, although we're not going to talk about it today, which is defined as the very severe loss of muscle mass that a lot of people over the age of 65 will go through. and that is very debilitating. And so muscle stem cells, just like a lot of H stem cells, come with a collection of intrinsic and extrinsic defects. When I say extrinsic, most of them can come from either the niche itself, which, as I showed you earlier, can be the supportive fiber itself, the ECM around the muscle, the nearby vessels, or the local factors, including other cells and receptors, as well as some factors produced by those cells directly. And you also have the systemic circulatory system, which you may think about, what's circulating in your blood and other tissues. So there are more distal. Of course, those things, those extrinsic and intrinsic changes are not happening separately. They often interact with one another. What we found, what I will talk about today, is how those systemic aging, including chronic inflammation, actually affect intrinsically those cells, which can lead to epigenetic alteration, DNA damage, accumulation of toxic metabolites, lipid peroxidation, etc. And usually the collection of those two things will lead to terminal fate, including senescence, apoptosis, lack of self-renewal, and just altering the fate of those cells in such a manner that they cannot contribute properly to what they're intended for, which is usually tissue regeneration or maintenance. So I always talk about this specific experiment that I think is one of the most critical experiments that was done in our field to demonstrate what I just told you, and that was done by Pro Munoz's lab in 2014. What they did here is they took cells from different mice at different age. So those are the age of the donor mice. They labeled them, those muscle stem cells, and then they transplanted them into a young injured host. And the idea here was to answer the question, if those cells are intrinsically bad, they won't function into a young environment, right? Because this mouse is young and healthy, and so it has a youthful environment that will alter the fate of those cells. Of course, as you can see here, the age of the stem cells actually impact how they contribute to regeneration. In adults, they contribute quite well, as you can see here with the GFP. It's a little reduced in old, but not dramatically. And then they come a point which is called geriatric, where the intrinsic hurdle that those cells go through are so far beyond that they are deemed irreversible. And this group actually called this geroconversion, which is an irreversible process where those cells go through premature senescence and cell arrest, and they just cannot contribute to regeneration anymore, no matter what the environment is. So there is a point of non-return. And so all of that to say that during my postdoc, I asked the opposite question, which is kind of more clinically relevant. I asked, what happens if you put some young, healthy stem cells into an old inflammatory environment, such as an H mouse? And you can see here during the transplant that adult to adult obviously contributed just fine. And then when you put those young cells into an H host, you have a dramatic reduction of how it can contribute to regeneration. And so now I have to explain the more complex layer of muscle regeneration that doesn't only include muscle stem cells. When it comes to efficient regeneration into an adult animal, you have first, and very importantly, the acute recruitment of inflammatory cells and molecules that will trigger a clearance of debris and a really pro-proliferative environment / activation of those cells, right? In my postdoc paper, we showed that in an adult scenario, immune cells such as macrophages or other immune cells can trigger the secretion of CCR2 ligands, including CCL2, 7, and 8, which will then bound directly to the stem cell, activating a phosphorylation event of p38 delta gamma, which in turn phosphorylate MyoD, the transcription factor, where when phosphorylated by this particular kinase, MyoD will sit at the TSS of myogenin and act as a repressor, which basically prevents the cell from going into premature differentiation. Think of it as a crosstalk between the stem cell and the environment, where the environment is telling the cell, now it's not the time to activate and proliferate or terminally differentiate, right? The environment is not ready for that. All right. In the age environment, what happens is you have something called instead of transient, a sustained or permanent inflammatory state where you have no resolution of the inflammation. The resolution of the inflammation is really critical for the progression of many processes, including myogenesis. And in this case, because the inflammation is chronic, you have this accumulation of both immune cells, but also all of the secretome that comes with it, including those CCR2 ligands. In this case, the CCR2 signaling is overactivated in aged muscle stem cells, which leads to basically the permanent inhibition of myogenin, and so prevent terminal differentiation, no regeneration. Of course, if you target CCR2 precisely when it's a problem, which is at the time of the resolution, not before nor after, you can actually promote myogenesis and aged muscle regeneration. And you can also restore the capacity for those young stem cells to contribute to regeneration in an aged host. So basically what we did here is we desensitized those cells to the inflammatory niche during the regeneration process of aged mice. That was just the introduction. Okay. And so the idea here is, following that, I wanted to understand exactly what was happening on the long term for those cells, seeing that aging is a slow process that slowly, change the transcriptome, epigenome, or proteome of those cells until it's too late and they can function normally. And so what we did here is we recreated an age environment into young mice by taking some aged plasma and chronically injecting that into the bloodstream of adult mice for 21 days. That alone was enough to reduce the number of muscle stem cells. So think of it, like this is obviously very indirect, right? Like you inject plasma into the bloodstream and that has a direct impact on stem cell in the muscle. That did not only affect, of course, the muscle stem cell number, but also their regenerative capacity. What you're seeing here is a cross-section area, so CSA in micrometer square, and it's the frequency of their size during regeneration. So the bigger they are, the better, right? And that's what you see with the saline here. And the mice that receive the aged plasma, you can see this leftward shift, which indicate that regeneration went actually poorly. And so because of time constraint, and I don't have time to really go through the whole thing, I'm going to skip and say that to link these processes directly to what was happening inside of the muscle stem cells, to link, the ligands that we think might affect the stem cell through the circulation directly, we did a collection of multi-omics that involved Luminex and single-cell RNA-seq and a lot of in silico assay using machine learning and AI. And at the end of the day, what we had was we linked this Go term, chromatin organization, as the most modified term in response to those cues, right? To age plasma injection. And among those lists, we have that stem cell marker PAX7, so that was kind of a nice and neat control. And then we found KMT5A. And at this point, nothing was known about this, and we were quite curious to understand how this lysine mediated cell transferase could affect the epigenome of those cells in response to inflammation. Okay, and so as you can see on this big graph where there's like a ton of like different methylation events, we are going to focus today on lysine 20 of histone H4, so H4K20, and particularly on KMT5A that happens to have many, many names for some reason, PR SET7, SETD8, SET8, and KMT5A. And so I think I'll just call it KMT5A if you don't mind. The nice thing about this, and that's pretty unusual in the world of epigenetics, is KMT5A, as far as we're concerned at the moment, has only one job, which is monomethylation of H4K20. And this one job is only being carried out by this enzyme, right? Which is different from other methylation mark on histones where you have 2 or 3 enzymes that can perform the same job. In this case, it's You only have this one enzyme doing this one catalytic activity. What's important to know too is this monomethylation is required for the subsequent di and trimethylation events by KMT5B and KMT5C, which means that if you delete KMT5A, which, spoiler alert, I will, you will not only lose the monomethylation of H4K20, but probably the di and tri, which adds a whole lot more complexity to it. As a general rule of thumb, even though it's not super studied, monomethylation is associated with open chromatin and transcriptional activity, while H3K20 trimethylated form is really heavily compacted chromatin, is usually constitutive heterochromatin, so really, really dense silenced region. So the first thing we did here, and that's kind of how I started this whole thing in my postdoc, was to check, okay, is KMT5A and H4K20 even affected in muscle stem cells? So we looked by Western blot and qPCR and flow cytometry, but I'm not showing this here. And we found that yes, like compared to adult age, in age muscle stem cells, KMT5A is down, as well as is main substrate, H4K20 monomethylation. And similarly, KMT5A expression is down. And so we wanted to basically confirm that there's a direct link between inflammation and the erosion of this mark. And so here, instead of repeating the process where we did chronic inflammation with the H plasma, we specifically targeted the ligand that we had previously identified, which is CCR2 ligand. And same thing, when we target CCR2 chronically, we can actually reduce the pool of stem cells, which leads to a defect in activation and regeneration. And we can actually basically prevent that partially by using some neutralizing antibody against those ligands. And to confirm again, yes, we did decline or decrease H4K20 monomethylation and the expression of KMT5A. So that was a neat way to kind of link those two together. So the big question here at this point was, what is KMT5A role in adult myogenesis and stem cell? So we generated knockouts for KMT5A in muscle stem cell specific mouse model. It's a PAX7 Cre ERT2. So it is a specific and inducible model by following tamoxifen injection. What we did here was I injected 5 times tamoxifen and then looked at the number of muscle stem cells either immediately after those 5 injections, a week after, 3 weeks, 6 weeks, etc., even 3 years. And what we found was that over time, you have this stochastic decline in the number of muscle stem cells, which was a problem, but I will mention that later, I guess. Okay, so now we established what was the role of KMT5A at homeostasis. It's important for quiescence and maintaining the pool of stem cells. What about adult myogenesis? To study adult myogenesis, or basically muscle regeneration, there are several ways to cause injuries, especially in rodents, including mice, which is what we use. You can use freeze injuries or different toxin, or salt, which is what we use in the lab. We use barium chloride. You can see here that rapidly after the intramuscular injection of this salt, you have a heavy phase of degeneration, which is the pro-inflammatory phase, followed by the resolution or pro-regenerative phase, which comes later around 7 days of injury. And what you see at the end here is a perfectly fine regenerated skeletal muscle. And a hallmark of this successful regeneration is centrally nucleated fibers, as you can see here. Again, same thing. We did this time the injury in the wild type and the knockout, 3 weeks or 6 weeks post tamoxifen injection. And if that's not clear for you, what you can see in the wild type is a very well regenerated muscle, where you can actually see, again, some stem cells, which is encouraging, right? You do want your pool of stem cells to go back after regeneration. And in the knockout, whether it was 3 weeks or 6 weeks later, at 21 days post-injury, regardless, what you're seeing here is essentially a bunch of ghost fibers, debris, and immune cells, basically saying that no regeneration occurred at all in those mice. And we found essentially no surviving muscle stem cells. So we started to be curious about how KMT5A was regulating the fate of those stem cells. And so we turned to transcriptomic methods. We used a collection of them, single-cell RNA-seq, bulk, and also something called precision run-on sequencing, which, if you're not familiar with this, basically what it does is that unlike bulk RNA, which really captures the entirety of the mRNA present in your cell, it will actually quantify the expression of nascent RNA, right? So it will capture the genes that are actively being expressed rather than just the mRNA that are already in your cell and might have been there for a while. There's two sides to it. The idea is not only you get what's actively transcribed, but you also get precisely at the nucleotide resolution, where is your polymerase II at the moment where you perform the sequencing. And so when we did look at the PRO-seq data from wild type and knockout, what we found was notch signaling. And you might know that or not, but in stem cell, notch signaling is notorious for regulating quiescence. And so a disruption of notch signaling would basically means, yes, like we are disrupting quiescence. And so in muscle stem cells particularly, notch is an important regulator for the fate decision at homeostasis when those cells are actually quiescent. You can see that notch is high. And as those cells activate, notch goes down. And then they have this choice that, like I was saying earlier, between going back to being a stem cell and quiescence or simply becoming a myoblast and differentiate. So we compared our RNA-seq data, PRO-seq data, and the pausing index, which essentially to say that if you're not familiar with what pausing is some genes, not all of them, are actually regulated this way. It's kind of a fine-tuning mechanism, and it's still unclear exactly why it's that way, but that's kind of what we're studying in the lab. So what you see here is the pre-initiation complex that binds the TSS. If the gene is regulated by pausing, what you're going to capture on the PRO-seq is this very peculiar profile where you have stalling of the pre-initiation complex, maybe 50 to 250 base pair away from the TSS. That pause can either go toward release and successful transcription with elongation and termination of the transcript, which is what you see here, or it can be stalled and basically the complex just come off and the gene is not expressed, right? And so we basically correlated here the fact that when those genes have a lower expression, they also have a higher pausing index, suggesting that for most of those genes, pausing is a way to regulate notch in a KMT5A dependent fashion. At the bottom over there, you can see RBPJ. For people knowing notch, it's kind of the main target, like one of the most interesting one. In the case of RBPJ, it's really one of the most important cofactors that help with activating notch genes at the enhancer of those target genes. And it was shown before in muscle stem cells particularly that when you knock out RBPJ, those cells basically straight up exit quiescence and go into terminal differentiation, which is kind of what we thought we were seeing. So we confirmed that in the knockout, we have less KMT5A and H4K20, makes sense. And we have also less of that RBPJ kappa, which is a notch coactivator. This is just mRNA expression for RBPJ. S and A1 are target genes of RBPJ and notch1. And so they are out as well. They're down as well, confirming what we thought. And this is just a cheap qPCR where you see KMT5A binding at different loci in the wild type, knockout, and the IgG. Just to say that, yes, KMT5A actually binds those sites. And the next question that I asked was, is this catalytic dependent, right? Is it just KMT5A? Is KMT5A catalytic activity required to regulate notch signaling? And so in this case, we used a selective substrate competitive inhibitor, UNC0379, which I will call kindly KMT5A inhibitor. And the idea is because it's a substrate competitive inhibitor, it doesn't bind or prevent binding of the SAM, but only of the substrate pocket in this case. So you don't lose any level of KMT5A per se, because that's not what you're targeting, but you are interrupting the de novo methylation of H4K20 monomethyl. And so same thing here, we showed that some myogenesis genes aren't affected, which really means that KMT2A is not directly regulating myogenesis, but really is regulating Notch genes specifically, including S1A1 and RBPJ. And so at this point, we thought that maybe reactivating the Notch signaling will actually prevent the loss of the quiescence. And so what we used here is we used a mouse that overexpressed in the Troza locus the intracellular domain, or Notch ICD, NICD. So you can see here the simple wild type, single knockout, and the double mutant that has both the KMT5A fluxed, and so it's a knockout in muscle stem cells only, and also the overexpression of NICD, same thing, only in PAX7 cells, muscle stem cells. So this is here the number of muscle stem cells, which was rescued by the overactivation of Notch signaling. And we basically restored quiescence. What you see here is Ki67, which is up when you do the single deletion, and back down when you do the rescue. And Ki67, if you don't know, is a marker of cell cycle entry. So if you're Ki67 negative, you are out of the cell cycle. Quiescence. We did restore the expression of those Notch genes, which makes sense. Unfortunately, what we did not prevent is the loss of muscle stem cells or restore their capacity to contribute to regeneration. You can see here again, wild type, knockout, and double knockout. No stem cells contributed to regeneration, and there was no surviving cells, whether it was in vivo after regeneration or even in culture. Those cells basically died after a few days. And this is when we started to turn towards single cell seq, because of course at that time the big puzzle was, because those cells die in a stochastic way in vivo, it's really hard to catch, right, what happens to them. And the idea is at any given point, when you take a mouse and you harvest those cells, those knockout cells, you are essentially isolating the surviving cells. So do they have the same problem that the cells that actually died? It's kind of hard to catch. And the single cell, hoping that we would actually see some fate trajectory here. And so we use wild type, knockout 2 weeks later, 6 weeks, or even 2 years, which is the blue, but I didn't really yield anything. Interesting. And what you can see at the top of this analysis here is a term called ferroptosis. And I make that joke every time I present this presentation, but it's also true. I had no clue what ferroptosis was when I found it. Luckily, somebody in my department back then knew about it and was telling me that I just hit the gold mine or whatever when he looked at those genes. So I believed him. So I looked into it, and ferroptosis, you can see it here, is a different form of regulated cell death that is iron dependent, and ferroptosis. As you can see on this graph, there's basically 2 arms to it. The first one is intracellular import of iron, which you really need for the cell to perform the Fenton reaction, which obviously has a purpose, not just causing havoc. But this reaction does cause the accumulation of toxic metabolites, including lipid peroxidation, which is quenched on the other side here by a selenoprotein called GPX4, and prevents the cell from dying through ferroptosis. Although this form of cell death was basically coined and discovered in 2012, so it's still been more than 10 years. Geez. It's fairly new, but the field has been very active into determining exactly what's the best method to decide what is a ferroptotic cell versus what's not, right? Because obviously you don't want to have any confusion. And you can see that when you look at those markers, which is lipid peroxidation, mitochondrial dysfunction, some gene expression changes, including in those ones over there, and TFRC1 mobilization at the cell surface, you basically hit the home run. Those cells are dying from ferroptosis and not from apoptosis, that have none of those things. And so we started to look into the knockout again. We looked at GPX4 localization, and what we found was in the wild type, GPX4 was actually rarely distributed in muscle fiber, but in the knockout, it was always near a iron-rich region that often was around the PAX7 cells or a muscle stem cell. So that got us kind of wondering, why is iron around the stem cell? And so we looked closer, and what happened, we found that in the knockout, those stem cells had themselves a ton more iron intracellularly. And when we quantified that by elemental mass spectrometry, we found that the order of magnitude is actually, in the thousands fold, where the wild type have very low amount of iron while they're quiescent, but the knockout had like, a lot more, which can be healthy considering how oxidative it is. And so we looked at lipid peroxidation, and as you may imagine, as we induced the knockout, and that was a time-dependent matter, and it kind of mimicked the way that those cells would die, we had an increased amount of lipid peroxidation that matched the cell death. So the model at the time, based on our transcriptomic data, was, well, in the knockout, we have, more of the receptor that puts the iron in, less of the transporter and the exporters, because their expression are down. So we have more accumulation of intracellular iron in those cells. And we also have less GPX4, which is what we showed right here. I guess it's the next slide. Of GPX4. And so that means more lipid peroxidation and ferroptosis. All right. And so we basically looked at how exactly KMT5A was regulating GPX4. When we re-express KMT5A, we do restore the expression of GPX4, suggesting that they are KMT5A regulate directly GPX4 expression. We found that yes, KMT5A bind at near the TSS of GPX4, only in the wild type, but not in the knockdown, and we found that it's restored, of course, in the overexpression of KMT5A. Kind of worked as a rescue experiment, and this is for H4K20 monometal. So of course, it's same pattern where you have the accumulation of H4K20 throughout the gene body. That's lost with KMT5A is down. Same thing, we wondered, is GPX4, the loss of GPX4, the only thing that actually causes ferroptosis? Of course, we can't be sure because we haven't done that in vivo and only ex vivo, but when we do re-express GPX4 in cells that are targeted with the catalytic inhibitor, we are restoring GPX4 expression, of course, in the overexpression of GPX4, but also we are restoring in those cells the survival of those cells, as well as reducing lipid peroxidation, right? So it makes sense. That was pretty expected that when you overexpress the one protein that does that job, you are going to fix the problem. All right, back to aging now. And so what is KMT5A role in muscle aging? What might the problem be here? So here we turn to a single cell RNA-seq again, as well as cut and tag. For those of you not familiar with cut and tag, it's the newest, fancier version of chip sequencing, also way easier and cheaper. If you have questions about that, I'm happy to talk about it after this talk. And of course, single cell RNA-seq. You can see here in blue are the wild type, and in orange, into those two nice clusters are the knockouts. And this is the 2D plot for the H4K20 monometal signal over the TSS of every single gene. So this is what cut and tag gives you. It gives you the signal or the map, if you want, of H4K20 monomethylation over the entire genome. All right. And so here, what we found when we looked at single cell transcriptomics was that, of course, the age cells really cluster on their own, and they kind of took on two different cluster or two different trajectory. And while I won't go into too much detail, what was interesting here is that if you look at cluster number 2 versus cluster number 3, one is what we would consider in our field the really canonical cluster of aging, where with the typical issue with cell cycle and myogenic gene expression. Well, the other one was kind of not known, and when I looked at the gene that were different in that cluster, ferroptosis showed up, as well as coronavirus for some reason. But I think it just shows up everywhere. And so, yeah, this is what allowed me to do this clustering analysis with cluster 2 and cluster 3. Anyhow, and so again, we looked at cut and tag. what you might notice is that the difference in H4K20, genome-wide between young and age is not that dramatic. Bear in mind, though, that there's only 1/10 of your muscle stem cells that are remaining in those age mice, and again, you are capturing the surviving cells, right? So the idea here, or our hypothesis, was that either this mark is age resistant, which we found to be not true, or what you can only capture the cells that lost enough H4K20 to still be alive, and obviously if you lose too much of it, you just die, and that's kind of the balance and the key trigger here that we're still trying to figure out, because we don't know what triggers exactly that loss of H4K20 monometal. So to confirm that ferroptosis was a problem, we looked at different fates that those cells could take. So here you have different markers through flow cytometry of adult and age muscle stem cells. We looked at senescence, apoptose, and ferroptosis, as well as basically others, right? Like those are dead cells that didn't go through all of those things. And when you look at all the cells that took those fates, you notice that the vast majority, not the vast majority, majority of them, 42% of them were actually ferroptotic. We actually sorted those cells for subsequent molecular analysis. So that means that at this point, they're already like lipid peroxidation high. GPX4 was indeed down, as well as KMT5A. So that kind of goes with our hypothesis. HMOX and PTGS2 being up are just essentially markers of ferroptosis at this point. They also had a lot more iron. Of course, it wasn't as dramatic as in the knockouts, because those are just H cells, but still a very significant difference in terms of intracellular iron accumulation. And same thing for lipid peroxidation. They were just more sensitive to lipid peroxidation as a whole. And that was even more true when we put them in culture. All right, so at this point, what we found was that chronic inflammation is directly linked to the loss of muscle stem cell number, that the loss of H4K20 monomethylation led to the loss of muscle stem cell, and ferroptosis caused this, right? And we kind of established at this point that H4K20 monomethylation, this decrease, is linked to ferroptosis and the loss of muscle stem cell. What we haven't linked yet is those two things, which is how I started this talk about inflammation. So can chronic inflammation, or as many people call it, inflammaging, cause ferroptosis in those stem cells? And so we went back to our model where we took aged mice, collected their blood, isolated their plasma, and chronically injected the plasma into those young mice to create this chronic inflammation model. Again, PAX7 number goes down. This is rescued by neutralizing antibody. H4K20 goes down, rescued. Lipid peroxidation goes up. We kind of knew that. And then it's rescued by neutralizing antibody. And even at the molecular level, intracellularly in those muscle stem cells, you can see that KMT5A is actually pretty suppressed or silenced in response to that chronic treatment, which is also rescued, as well as GPX4 expression. So there was kind of a causal link here between chronic inflammation and the regulation of ferroptosis in those mice. So we went a little further and again did this more specifically with CCR2 ligand, which you've seen those data before, where you have this decrease of H4K20, really linking CCR2 activation to the erosion of this mark. And here we show again that GPX4 is down, HMOX is up, so those cells are basically ferroptotic with high lipid peroxidation. And when we do the same thing in culture, those cells actually show signs of dying during chronic exposure to those chemokines, which is rescued as well by the reintroduction of neutralizing antibody. Okay. Now, that was the craziest experiments I think that I've designed during my postdoc, which took me nearly 2 years to complete. It was a crazy gamble. What I did was I took a small anti-inflammatory drug called Binderit that instead of targeting the CCR2 receptor like I did during my postdoc, was targeting specifically the transcription of CCL2, 7, and 8, so the ligand themselves. And that really led to minimal interference with immune reaction, right? Because it was basically just keeping everything low instead of really inhibiting the entire pathway. And so those mice, just wild-type mice, were basically treated weekly with this inhibitor for 12 months of their life, from 12 months on to 24 months of age. This is just a confirmation that the anti-inflammatory drug works. We found that after 12 months of treatment, the amount of circulatory monocytes or lymphocytes change, right? Like you have less monocytes when you compare it to, I don't know why the legend is missing on this over there. So that's just like, adult age treated with vehicle, so nothing. And the age mice that were treated for 12 months with the inhibitor, they have less monocytes, they have more lymphocytes, less neutrophils, so less pro-inflammatory phenotype. And when it comes to protein itself in circulation, so that's in the plasma, you have less CCL2, less CCL7 and 8, as well as less TNF alpha and IL-6, meaning that basically by targeting those three ligands, you just didn't decrease their expression, but also overall the inflammation or inflammaging in those mice by just preventing a chronic inflammation from arising. And so functionally, that led to better regeneration, an improved number of muscle stem cells, whether it was before or after injury, better strength recovery. So it means like you did an injury and then we looked at their strength, and they were stronger in the mice that were treated. You can see here, this is again the size of the fibers. So you can see that in age, most of those fibers are pretty small, but from the age that were treated, you have this rescue, and so it's just shifted to the right because those fibers are bigger. Inside of muscle stem cells, we also reduced the lipid peroxidation level back to basically basal level, as well as prevent this accumulation of iron intracellularly, and restore the amount of the expression of CAMT5A, which was just a cherry on top of the cake at this point. Not super necessary, but I still insisted on doing that for revision purposes. Here, we basically decided to check whether those benefits were transferable. So bear with me. Here, we took the plasma of age mice, regular age mice, or the plasma of age mice that were treated for 12 months with Binderit, all right? So basically, they don't have inflammaging. And now we chronically injected that plasma back into young mice. In the case of regular age mice, you obviously create what we've seen before, which is chronic inflammation, and you reduce the amount of PAX7 cells, H4K20, regeneration, and all the other aspects that we just discussed. When you take the plasma of those mice, those age mice, they're 24 months old or more, and chronically inject that in adults, you basically just rescue, number of stem cell regeneration and ferroptosis as a whole. And so that's kind of the model. I didn't say that, but yeah, most of the data I showed today were just accepted for publication in Nature Aging, except the nudge data. Those were not. So this part here is not there was just unpublished data. So what we saw here is that really CAMT5A is repressed through systemic inflammation, which can prevent with the addition of Binderit. And that affects, of course, the fate of muscle stem cells over time by eroding H4K20 monomethylation, which is critical to maintain an anti-ferroptotic transcription. Thanks to my lab, my canine mentoring team, my funding, including all the institute here that supported me since I joined. And thank you for your attention, and let me know if you have any questions. I don't know how I did on time because I started this Zoom like a long time ago. So, yeah. Okay, I'll start by answering your second question because that's the fastest one. Yeah, actually, those cells do kind of like rejuvenate in like so long as they're not geriatric to begin with, they basically hit a hard reset. It's not fully understood because nobody has done really super long longitudinal studies, but what's been published actually like really recently, I think like a few months ago, they transplanted H cell into a young mouse, and then they isolated the cells back to do something called SMART-seq. So it's like a really small amount of cell sequencing. And they found that, yeah, like the transcription pattern signature of those cells is very close to a young cell. They also function well because you can do serial transplants and they will do the same job over at least 3 transplants, as far as I know. How long does that last? I don't know. But yeah, like, you know, the best clue we've had a long time ago and how this whole thing started with rejuvenation and you know, extrinsic versus intrinsic was started by groups that did heterochronic parabiosis, which is basically linking the bloodstream of 2 mice together with the surgery. And if you make a young mouse and an old mouse together, right, like they just share the bloodstream. So now technically all the age factors and cells go to the young mouse and vice versa. And what they found was, yeah, like exposure to young blood rejuvenated those cells in the aged mice. But the opposite was true too. Exposure to that aged blood, those age factors and age cells really prematurely aged that young mouse. And so this is kind of how this field like blossomed at this point. And I forgot your first question. Oh, yeah, the CCR2. So in the case of homeostasis, we don't really know. As far as I can tell, and that's something that we are interested in, it seems that the accumulation of immune cells, you know, it's not a lot, obviously, inside of the muscle, but they seem to be more resident derived, like they don't have those recruitment markers. If we do an injury, this aberrant chronic inflammation that ensue is due to recruited macrophages. Most of them are actually bone marrow derived. They have the whole CCR2 patterns and everything. But when it comes to chronic inflammation, that is not really resolved. We don't know, right, what leads to chronic inflammation and aging. A lot of the clues, and that's one of the part of the thing of what I'm studying, is looking at bone marrow specifically and hematopoietic stem cells. The current idea or hypothesis here is some event triggers in the bone marrow where hematopoietic stem cell go through clonal expansion. This clonal expansion tends to be more myeloid prone. So they will give rise to more myeloid progenitors, more pro-inflammatory progenitors. And so that's called something that's called myeloid bias. So that's why you have less lymphocytes and more myeloid cells. Because those cells are more pro-inflammatory, you can imagine that they might produce more pro-inflammatory cells in circulation, which will then lead to clonal expansion to expand even further because those cells now how to resist this pro-inflammation in their niche. So they will give rise to more myeloid cells. And it's basically a loop until you essentially just die, right? But yeah, so this interaction between the aging of the bone marrow and hematopoietic stem cells and how that systemically affect other organs is what is part of my grants, actually, part of my funding with the NIA, and try to understand how that, yeah, impact each one another. Yeah, no problem. Yeah. Yeah, so the first question, so do you ask about how iron is important, metabolism is regulated in those cells? That's something I'm studying right now. We don't know. We don't really know why in the first place quiescent stem cells would even need to have iron to begin with, right? Because like iron, as far as I'm concerned, is mostly involved in mitochondrial function and processes with metabolism that requires the cells to be active. But the definition of metabolism is of quiescence is that they are low metabolic activity or none. Obviously not none because they still need to be alive. So how it's regulated, why it's needed, is something I'm looking into right now. Um, it's it's very surprising as I went into this field how little we know honestly about iron, and especially in stem cells. It's pretty crazy. Like, yeah, like we know it's the most abundant, element or metal in the body, and it's required for like a lot of processes, but exactly what it does is not really known. Something that really triggered my curiosity though is when I discovered that iron was used as a cofactor for the metylase, specifically Jumanji containing the methylation. So there's a direct link between epigenetic regulation and iron level. If you treat your cells with iron, a lot of the time what's going to cause is going to cause hypomethylation of a lot of epigenetic mark or even DNA because that enzyme require iron. So this kind of loop where epigenetic might regulate iron and iron regulate epigenetic level is something that I'm interested in at the moment, following up on this paper. The second question, yeah, I think there's a lot of applicable application for this. One of the avenue we're looking at right now with people that work in the blood center is all the people that do receive transfusion and or bone marrow transplant. What's the relationship between the aging trajectory or the age of the donor and of the recipient? How that might affect the transplant efficiency, survival rates. And so we're looking at things like, of course, leukemia, MDS, and different diseases in a more scary manner. And I'm not the first one to show that, like I was saying before, because of heterochronic parabiosis. A lot of people have looked into, hey, what if we sell young plasma as a rejuvenating cure, right? Like cure for aging. We'll just collect plasma, which a lot of companies in this country are doing, and most of the time it's for good use, like making coagulation factors and other things. But there's a lot of people, especially in California, that are like biotech that are selling you young plasma with the promise that they will reset your stem cells in your body. All right. So it's obviously not new. Like in Switzerland, they've been doing that with young goat serum for decades, which yeah, so rich people in Switzerland, get frequent goat serum injection, I guess, which is very strange, but yeah. Yes. Not that I know of. Yeah, not that I'm aware of. Yeah. Did you have an idea in mind for asking this question, or? That's a good idea. I don't think so, but I don't think we know at least. I know. So actually, it's funny. Right before I got this, like maybe a month before I got this result, a Cell paper came out about how hematopoietic human hematopoietic stem cells are very sensitive to ferroptosis, more than any other cell death that we know. Exactly. So it was really cool, like to, hey, I guess we're on the right track. There's something there. To be honest, I think I just got lucky because the term in the KEGG pathway was added, ferroptosis, in 2021. And so if you had done this exact single cell or ProSeq or whatever in 2020, the term did not exist. It would just have never showed up in the list. So, it's not that I'm the only one that I've ever found this. It's just that I happen to have the data at the right time, which, it's definitely, like, I mean, of course I wondered, right? Like when I found that and it was the top term, like, why did I hit this and nobody else before? That makes no sense, right? I'm not the first one to do single cell sequencing. That's it. That was just like the term was not there when all the bigger lab rushed to do their own single cell seq whenever the single cell RNA seq was like very popular. So sometimes serendipity is the thing. Yeah, guess. Keep up. Yeah, yeah. It's yeah, it's true. Okay. Well, thank you for your questions and your attention.